Welcome to another video. This time we're looking at Chapter 5, Part B, the structure of DNA. This multi-region figure shows how the building blocks of DNA are brought together inside cells. This would represent an energy requiring anabolic reaction, and it also has greater order, so the entropy of the system has decreased. The nucleotides are first assembled in the cytoplasm utilizing food molecules, sugar, phosphates, and bases. Those are then polymerized to form a strand of DNA, and that strand of DNA is then combined with a second strand to make the DNA double helix. The two strands are held together by hydrogen bonds, whereas the molecules making up each strand are held together by covalent bonds. The only way the two DNA strands can adhere together in any semblance of a structure is if one strand is facing north to south and the other strand is orientated south to north. This is called the anti-parallel arrangement of the two strands. We'll address this again shortly to be understood. One of the most important is the width of the molecule itself. The two strands coming together have a defined width. This is obtained by utilizing a base with two rings alongside a base with a single ring. So the total number of rings in the bases from one backbone to the other is going to be 3. So if the student examines figures of DNA throughout the book, they will realize that this pattern exists every time. This is only possible if a purine with 2 rings is bonded opposite to a pyrimidine with 1 ring. So adenine can pair up with thymine, and guanine can only pair up with cytosine. The relationship between adjacent bases is irrelevant in DNA because information can be stored in any order. However, for each rung of this ladder, the relationship is fixed. So here, adenine only forms 2 hydrogen bonds with thymine, whereas guanine always forms 3 hydrogen bonds with cytosine. This has to do with the distance the hydrogen atoms are away from their counterparts across this minor gap in the DNA helix. Of course, natural DNA does not exist as a 2-dimensional molecule. It exists as a 3-dimensional spiral. The distance between the 2 anti-parallel strands is not even. This generates 2 grooves which run around the molecule. A minor groove separated from a major groove by 1 backbone. These major and minor grooves are therefore alternating up and down the DNA helix. This is a very important concept to understand because molecules like proteins, which need access to the DNA using their molecular shape, can make contact more inside the major groove than inside the minor groove. Indeed, the bases deep inside the DNA helix are accessible more from the major groove than they are from the minor groove. These concepts will be built upon later in this course. Based on calculations of natural crystallized DNA molecules, we have calculated that the distance between adjacent bases in the DNA molecule is 0.34 nanometers. This means that along the DNA helix that there are 10 bases for every helical turn. This is a fixed number which is hard to modify. This figure further addresses the orientation of the 2 DNA strands. If you look at the strand on the left-hand side, even though it's made of 4 nucleotides in this very simple example, it still has 2 ends. The top of the molecule has been labeled as the 3' end, basically because the sugar molecule has a free hydroxyl group on its 3rd carbon, whereas the other end of the molecule has been labeled 5' because the sugar molecule has a carbon atom to which the phosphate group is attached, and that carbon atom is the 5th carbon atom on that terminal sugar molecule. For the other strand, the reverse situation applies because that strand runs in an anti-parallel direction, as we discussed earlier. Therefore, the 5' end of that molecule is facing north, and the 3' end is facing south. Following the same logic, this sugar molecule at the bottom will have a free hydroxyl group on its 3rd carbon. Likewise, the top sugar group will have a carbon atom attached to the phosphate, and that will be the 5th carbon atom. All linear DNA molecules therefore have both a 5' and a 3' end on one side and a 5' and a 3' end on the other side. As we'll learn in Chapter 6, DNA can only grow in one direction. Now that we have a decent understanding of the structure of DNA, it's time to move on to its function. It is commonly perceived that information in DNA is used to make proteins. So in this example here, there will be a segment of DNA which we call gene A that will encode some messenger RNA in some form, and that messenger RNA would then be translated into a protein. Another gene somewhere along the DNA would do the same, and so would other genes also situated on the DNA. Well, that begs the question, what is a gene? The answer depends on which type of textbook is utilized. Biochemistry textbooks approach the answer from one direction, whereas cell biology textbooks approach the answer from a different direction. Regardless, this definition of a gene is most acceptable in all cases. So a gene could be defined as a locatable region of genomic sequence corresponding to a unit of inheritance, which is associated with either regulatory regions, transcribed regions, and or other functional sequence regions on the DNA. This may seem like a handful to remember, but over the course of the next few chapters, it will become clearer as to why such a complicated definition is necessary to address all genes. The student is asked to dwell on this definition until they understand the appropriate meaning. Unfortunately, there are major differences in the organization of information along DNA and chromosomes between prokaryotes and eukaryotes in particular. The next section applies mostly to eukaryotic gene organization. When we examine the genes of most eukaryotes, there is an amazing pattern which emerges. The pattern suggests that only tiny proportions of the DNA actually code for anything in particular. There are many regions inside the gene itself which are first removed before messenger RNA is produced. These regions have names. The parts of the gene which are kept and therefore encode amino acids eventually are called exons, indicated in green, and the regions which are removed, the intervening regions, are called introns, indicated in this figure by the red arrows. As you can see, large chunks of the DNA are removed and only small chunks of the gene are kept. Introns are not found in prokaryotes, so this is only relevant to most eukaryotes, with a few exceptions. Most of the DNA in a eukaryotic cell is located within the nucleus. There is some DNA in other organelles like mitochondria and chloroplasts, but the vast majority of the DNA is in the nucleus. The membrane surrounding the nucleus is a double membrane consisting of two lipid bilayers with pores in between, allowing material to be exchanged between the cytoplasm and the nucleoplasm. The structure of the nuclear membrane is very complex, as indeed is the organization within the nucleus of all the material that's kept in there. This slide provides a better representation of the organization of a eukaryotic nucleus. As we can see, the inner surface of the nuclear envelope is coated with special proteins, which are known as the nuclear lamina. The nuclear lamina is a bridge connecting the nuclear membrane to the chromatin, within the nucleus. The organization of the DNA within the nucleus can be subdivided into two types: chromatin, which is loosely packed, versus chromatin, which is tightly packed. So heterochromatin is tightly packed DNA, and that is found adjacent to the nuclear lamina. Other DNA, which is loosely packed, is found in the interior of the nucleus, forming various structures. One of these is the nucleolus, and as we alluded to in a previous video, this is the site where many chromosome arms, which are encoding proteins and RNA molecules for ribosome construction, are located. One terrific way of identifying the location of chromosomes is using modern technology, where different substances, called chromomeric substances, can be used to label each and every chromosome a particular color. The computer then rearranges the chromosomes according to those colors and their size. Any picture of chromosomes is called a karyotype. So here in panel B, we have a chromosome karyotype showing the arrangement of chromosomes from a human male. All these chromosomes were recovered from a single mitotic cell nucleus. More sophisticated chromosome staining techniques using different substances enable us to label regions of the chromosome with different densities of those particular substances. In this case, something known as G banding binds to regions of the chromosome which are rich in adenine and thymine bases. And you can see these regions are scattered on every chromosome. Professionals who are appropriately trained can identify mutations in chromosome locations and segments. These large-scale chromosomal rearrangements are known as chromosome translocations. Indicated in figure A, we see here on the left-hand side a pair of normal chromosomes, and adjacent to them a pair of chromosomes which have undergone a type of translocation. Figure B shows us how that can be depicted as a cartoon. The purpose of this slide, taken from a real yeast chromosome, simply suggests to us that since there are two strands of DNA, either one could be classified as carrying the information of a gene. There is no reason or rhyme for this arrangement. In this particular location, from a yeast chromosome, the top strand carries very few genes, whereas the bottom strand carries a large number of genes. Also evident are the gray areas, which do not code for anything in particular. Now, what you're not seeing is that within these genes we do have lots of introns, but those have not been colored in this particular figure. Further confusion may lie in understanding the number of chromosomes between related species. Again, there is no simple pattern. Here we see two types of goat taken from neighboring countries, China and India. So these goats are relatives of each other in terms of an evolutionary perspective. Looking at their chromosomes, we can see that the chromosomes of the Indian goat are very intact compared to the chromosomes of the Chinese goat, which are very fragmented and very short. So the number of chromosomes in one species is much greater than the chromosomes in the other species. Perhaps the most revealing slide in this section of the chapter is illustrated right here. Humans have chromosomes that range from large to very small, and the numbering system follows the size. So number 1 is the largest, and chromosome number 22 is the smallest. As indicated here, this is a representation of the smallest human chromosome, chromosome number 22. So chromosome number 22 can be magnified through a number of steps to reveal the arrangement of information along its length. In panel A, we have the entire chromosome, 48 million base pairs in length. If we take a small segment of that and magnify it, as in panel B, we find that about 10% of the chromosome arm contains about 40 genes. And those genes are scattered along both strands of the DNA. Magnifying a small region of that to give us 1% of the chromosome, we find that there are 4 genes in that location. Zooming in on one of those genes illustrates further how the introns are arranged between the exons. Every gene has a regulatory region, a switch that turns the gene on and off, and that is indicated here in yellow. Thus, vast regions of the human chromosome are geneless. They may have other functions that we have not yet discovered, but as far as genes are concerned, they don't contain many in those locations. Based on the latest understanding of what different parts of our DNA do, we can come up with a diagram such as this. The top bar represents all the DNA along all our chromosomes. If that DNA is then classified into different types, we find that only a tiny fraction, about 2%, is coding for genes. So only 2% of our DNA actually results in the production of messenger RNA, and that messenger RNA then codes later on for proteins. That is a tiny amount. The introns represent a further 20% or so. So only about 22% of our DNA can be recognized as being part of genes. What about the rest? We believe that a further 30% or so is responsible for controlling the genes within our DNA. So that could be controlled, that could be known, that is referred to as controlled DNA, which still leaves about 50% of our DNA. And most of that is repetitive DNA, has no known function, and we'll talk about that at a later date. Another form of analysis that we can conduct is to compare our chromosomes with other species. In this figure, we're looking at human chromosomes, one of them, and we're comparing our chromosome to that of close relatives, the orangutan, and that of the lemur, a more distantly related primate. We can see that compared to the lemur, there have been some changes in the orangutan and further changes leading to the production of the current human chromosome. Analysis of this nature reveal the haphazard way in which genetic material can be reshuffled and lead to the present configuration. Finally, for this particular video, we end with this slide showing how we can combine information from multiple chapters to support the current understanding. Here we have the cell cycle, where cells go through various stages of manipulation of their genetic material. We can see that in interphase, the chromosomes are long and thin, barely visible in the electron microscope, but invisible in the light microscope. The nucleus is intact. During the S phase of the cell cycle, the DNA is duplicated, and we'll cover that in chapter 6. Once the DNA is duplicated, the cell enters mitosis, a particular phase of the cell cycle. Here, the chromosomes are manipulated and segregated so that one half move in one direction and the other half of them are moved in the opposite direction. The cell then divides down the middle into 2, and we end up with 2 daughter cells. These maneuvers are very demanding on the cell and take lots of energy and coordination. Therefore, one can understand when things go wrong, the cell either commits suicide or becomes cancerous.